to the waiting room and get rid of that. So give me 1 second. Sorry. Let's see. Okay, maybe I cannot turn it off. I'll just keep my eye on it and go. Hi, Andrew. How are you?
Hi, I'm good.
What time of day is it for you, Andrea?
5 a.m.
5 a.m. Oh,
Where are you dialing in from, Joe?
I'm in my pajamas. Melbourne, Australia.
Okay. I'm trying to do the math. So I lived in California for a long time, and I feel like I used to be able to do the Australia to California math. And now I live on the East Coast, and I know it's only 3 hours difference, but now it doesn't work in my head anymore. Obviously, I'm so used to it. I forget, does Australia do a daylight savings time?
Yes, we just changed to daylight savings about a month ago.
Okay.
Yeah. It just made it earlier for me, I think. Yeah. But it's worth it to see your beautiful faces.
Oh, you are very nice. All right. Okay, we've got a bit of a group now here, so I think we can get started. I just have a few slides to share a couple of updates, and then we'll turn it over to Andrew, who is going to be talking today about gene therapy, kind of a 101, all the things that we should know. As you know, we launched our gene therapy consortium 4 years ago now. We have 8 different creatine deficiency gene therapy projects going on around the world. So it's of big interest. It may not be available today, but lots of interest and even a couple of things that I can share today. So one quick plug, if you're in the Bay Area or maybe you're traveling in January to the conference that starts on the 12th, we will be having a fundraiser on the 11th. And it is hosted by Ultragenics. They so very kindly invited us to be the beneficiary this year. And so if you are in that area, check out /a-rare-affair on our site. And we're really excited because we are devoting all of the funds that we raise from this to the 2026 symposium. So it's great. Also, we zoom out, and I'm really excited because, again, it means every dollar that we raise, we can funnel straight to research. And on that topic, I just wanted to call out, if you haven't heard, Holiday Heroes. We start it November 1st every year. So November 1st, until we say the Holiday Heroes campaign is done, every dollar we raise is restricted. It is to fund research. And we announced in the spring, Race for a Cure, call for letters of intent for research projects that are advanced and have an eye on the clinic. And we got all of those. We responded, and we're currently having presentations from researchers and reviewing and considering 8 different projects. And we've probably seen our very lofty, maybe even scary goal of $5 million to fund these grants over 2 years. And I want to share an update. We're actually at 1.5 million. So we are making progress, and this is a race that we can win. And for this reason, this is the most important Holiday Heroes campaign. We are focusing really on direct outreach this year, and we had a new board member join and do a direct email campaign to his close friends and families and their close friends and families, and that raised $500,000 of that dollar amount. So We have templates. We're going to help you as much as you'll let us help you. And I think this is really going to be an amazing year. And I also, in the templates, we have some details on what are these projects that we're trying to fund. They range from drugs that deliver creatine past the blood-brain barrier into the neuron based on the initial findings, 2 projects, and several gene therapy projects. There's mRNA and enzyme replacement therapy for GAMPT. So a lot of really, really exciting project proposals from honestly some really prestigious institutions that want to help and are committing to really aggressive timelines. So if you want to know anything else about Race for a Cure, I'm an open book, barring any NDAs, and I would love to tell you all about it and help your family set up this campaign. I really believe together we're going to do this. So, and with that, I just want to call out again, we have something called the Momentum League, and it's people who've signed up for a monthly donation, just to take it off their mind and to make a little bit of impact, to help ACD take their mind a little bit off of fundraising. And these families together are pooling quite a bit of support every month for us. And it's been really, really, really impactful and helpful for ACD on the back end, keeping things up and running. And so if you can set aside 20, 30, 40, whatever dollars a month, go to creatineinfo.org/momentum, and let's get you signed up and get your family on this wall of supporters. Next community chat won't be till January. We're not going to have one in December, and it will be focused on potty training in our population. Ashley Hickey is someone who's worked with a couple of patients and has amazing tips. If any of you have heard of other people in the community, like hiring a specialist, this lady's great, and she will help you be brave and believe in yourself and your child and get started if this is something that you're interested in. And so with that, I'm going to turn it over to our awesome friend, Andrew. And Andrew, I'll let you start sharing your slides.
Thank you. It's really nice to be able to be here. I appreciate it. I just want to make sure, can you hear me okay?
Yeah, great.
Okay, great. Awesome. So I have slides. I'll share those. But really, I think the most important thing, and the thing I'm going to ask everyone that's on this call to do, is like raise your hand, ask questions, interact. Like, I'm happy to be here throughout the whole time. My favorite thing to do is be able to talk with folks and answer questions. And there's a lot of stuff and a lot of unknowns when doing drug development. And I'm happy to be as helpful as I can be. So I'm going to see if I can share my screen. Give me 1 second here. Let's see if this works. Can you see my screen? Heidi, can I get a thumbs up? Yep. All right, awesome. So it stopped. Screen sharing has stopped because the window closed. Stand by. So I'm much better at things other than audio-visual systems. So stand by. We'll do this one more time. We got this. And it just keeps failing. Okay, I want to do this the old school way.
Andrew, I just want to say you've got me beat, because at least you're aware that you're trying to get your slides sharing. I think I wasn't sharing my slides that whole time, so I apologize.
Let's try this one more time. You see my slides. I'm sitting inside of PowerPoint right now.
Starting, and yep, I'm seeing your slides with the PowerPoint frame around them.
Let's see if it's going to play nice. Is it now set up as a regular presentation?
Yep, looks great.
Okay, so let's see if it plays nice. So, hey everyone, this is me when I have a better looking haircut. My name's Andrew Steinsapper, and it's a privilege to get to be here. I'm trying to think of when I met Heidi and when I first got introduced to this community. I think I got introduced to this community because I answered an email, I was getting ready for one of the biggest conferences in my field, and invariably I was doing what I always do, which is finishing things at the last minute. And I was sitting at my office on a weekend, and I got an email from a principal investigator or researcher who reached out asking if I had heard about this disease and if I'd be willing to talk about things. And I said, absolutely, I'm going to be at the conference. Let's talk, let's hang out. And they then responded back, just going back and forth, emails over the next 15 minutes, fast and furious. He said, like, would it be okay if I brought someone from the Patient Foundation? And I stopped emailing, and I was just like, yes, like, absolutely. I was like, how do I make sure that I get this across in words without sounding like a crazy person? But so that everyone knows, so my name is Andrew Steinsever. I've spent my career trying to do whatever I can to help move things forward in rare disease. And my favorite thing to be able to do is actually interact with patient communities and help center them and empower them and put them in the driver's seat for therapy development and make sure that they're always having a seat at the table. My background, I grew up in California. I was lucky enough to be born there, and as a result, go to University of California, Berkeley as an in-state student. So it was a little bit cheaper because I had to pay my own way. And I studied chemical engineering, and I eventually went out and started working in the gene therapy space because that's what I did as an undergraduate researcher. And as the normal joke that I tell, and I think Heidi has heard me say this before, is I wasn't pretty enough, nor was I smart enough to get into graduate school, so I had to go work directly. And I had the chance to work at a company called 4D Molecular Therapeutics. They're public now. They have multiple products in phase 3. I helped with setting that company up and moving things forward. I then worked at a consulting firm called Dark Horse Consulting that focuses in cell and gene therapy. During that, I worked with folks in venture capital. I worked with folks in pharma and biotechs and foundations. And then I worked for who is now my employer, Deerfield Management. Deerfield Management is an investor. They invest in public securities as well as private things, so doing venture capital. And I sit on a team internally. Part of my day job is running therapeutic programs, bringing them out of academia or coming up with new things. And then I also get to sit and advise a number of our gene therapy companies, including Apertura Gene Therapy, where I'm the acting chief technology officer. And I've stolen their slide deck for some of this, which is why you're going to see some Apertura branded things, but I have some cool data there. I've adapted, or I've attempted to adapt a presentation that I've given in the past that I think helps folks center and think about, like, what are we doing and why are we doing it if we're developing a therapy? And my thought process for this, and I sometimes get laughs when I say this when I'm at conferences, and sometimes I get people staring daggers at me. Even though my whole career is focused on cell and gene therapy, it is my least favorite modality. I don't want us to have to use it. I would much rather we have simpler things. I'd much rather we have a tiny little pill that we could take to fix things, or if it would just be a change in diet to fix things. But in some cases, for some diseases, it's unfortunately, there is no other option. And I think it's important that when we're doing drug development, when we're thinking about these things, and we're talking to folks living with disease and we're talking to their advocates, that we ask the question of what actually matters. What are you looking for to make sure that the things that are developed are what are needed, and we're not just developing drugs for doctors, but recognizing we're developing for people living with disease. So with that very long introduction, let's talk about, like, why we're here. The way that I usually do this is I'm talking to groups of scientists and researchers, and I'm at a conference and I ask, like, why are we here? Like, what are you doing here? It's not just because someone paid for your conference attendance. It's not because of the OPE coffee or the free food, but, like, we're usually here because we all recognize that unmet medical needs are opportunities. An opportunity to do something to try to help move things forward. And the thing I try to challenge people to remember is that even when we have approved drugs, there are still unmet medical needs. Folks, I've been a caregiver, and folks in my life, we thankfully had medicines that were approved for them, but that didn't mean that all their needs were met. So a clarion call and a reminder to at least these scientists and researchers that there is no such thing as done. And the way that we know this is we keep talking with and working with the communities that we're looking to support and understand where are we and how much further could we go. But with that, like, thinking about what it means to get a product approved is terrifying, because if you're starting saying, like, we are here, we don't have anything, and we want to get something and we want to be done, what does it mean? There's a longer set of presentations that I gave that are online and available through ASGCT talking about all of this. Each one's about 2 hours. And the back half is 30 minutes on each time of Q&A with both advocates and scientists and researchers. So I've stolen this slide from there. The way that I think about it for what is our goal and what we're doing, like I don't actually care about getting to first in human. I don't care about getting the product approved. The thing I want to see is the 1,000th person get access to the drug once it's approved, making sure that everyone has the ability to access this, that needs this. And so the only way I know how to solve problems, because I'm an engineer, is to try to work backwards. And I know that to get a drug approved, we have to have submitted certain things to the FDA that say that we did well in clinical trials. And in order to do that, I know that we have to complete those clinical trials. And in order to do that, I have to get something called an IND, an investigational new drug application, to be allowed, which allows me to do human studies. And I know that to do that, I probably need to do discovery. And if I kind of walk backwards there, I can then figure out that this is the area that we need to focus on if we're trying to get over here. And if we know that we're trying to get over here, we can think about how that informs the things that we'd be doing. I'll get to the gene therapy, I promise. When talking with folks, it's, I think, important to challenge, like, what are our goals and what's the biggest point or most important thing that we're doing? And when I've surveyed people, I usually get some version of these as answers. Some folks will focus on publication. Others say that we need to get data that's patentable, or we need to get the IND filed. And for me, the way I think about it is like all these are necessary. These are all steps along that way. And that's why it's important that advocacy groups help with understanding where the needs are, help develop animal models, or fund research, or move things forward. But like the real reason why we're here is to get that drug approved commercially so that everyone can have access to it, getting it to a point where, you know, we're in the final mile, and the thing that we're doing is making sure that insurance covers it or knows that it's important to be used. Where all of these other steps can help us get there, but they're not actually our goal. And as a result, like, we need to be looking much, 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 much further out. When thinking about that, then, and we know what those goals are, and our goal is to get that far, and the only way to do that is to go through it. I like to know what the rules are. And the reason why I'm making sure to share this, the rules, so to speak, are all publicly available. The FDA is a wonderful organization. That might sound for some folks like a surprise for someone that works in drug development to say that. The reason why I say it is because they're really good at putting out information about what is needed. This slide is adapted from information that's available online, that's been available for a really long time, about what's needed to be done in drug development. When I'm not doing my day job, I'm volunteering with different rare disease organizations, helping them think through strategy in how to both raise money and spend that money. And whenever it comes to thinking about drug development and funding research in academia and whatnot, I really try to hammer home that the way that we move things from an idea to in people is by answering these 7 questions. And that's it. These are the only 7 questions we have to answer. Now, this guidance is for cell therapy and gene therapy, but it actually applies across the board. If you go look, there's a similar one for doing small molecules. And when it comes down to it, it's questions 5, 6, and 7 that a lot of rare disease advocacy groups will end up having a lot of information on, like understanding what the folks living with disease, what that looks like, what that means, what would be meaningful as an improvement. Number 7 is something that's actually true for all drugs. There's always this question about, if we're using this, is there a danger or risk to those around someone? Might be a little bit hard to conceive of, but if we think of something like radiation therapy or chemotherapy, where the stuff that you're taking when it comes out of you could also be dangerous, we always think about these to make sure that folks are safe. It's really questions 1 through 4 that we do the cool science, so to speak, where we're doing the experiments that say this molecule, this drug could have an effect. And these experiments are actually not that complicated. And you all have done such a wonderful job in standing up and making sure that you're counted and getting folks in the research community to spend their time and energy understanding these diseases, that we can say, oh, we know that when this gene is damaged. We don't make this protein, and as a result, we have this disease. And so when we're talking about biological rationale, at least in gene therapy, in some cases, my friends that I also work in other modalities, but my friends that work in other modalities will make fun of me and say that I have a really easy job because I get to say, look, that thing is missing. That's why they're sick. And if we work in a mouse or in a cell and we put it back in, maybe they're not sick anymore. And at a high level, like that is effectively true. And that's what we're doing in identifying the biological plausibility, in thinking about the active dose level and putting things back in, finding where we start, and then thinking about the feasibility of executing those studies. But if it's only 7 questions, why aren't we doing more of it? And I think a lot of time folks will use whatever tool they have at hand. If a lab is really, really good at using cell therapy, they're going to say the way to solve this is cell therapy. And if a lab is really, really good at adenovirus, they're going to say we're going to use adenovirus. And if a group's really good at AAV, they're going to say use AAV. And this is a part where it has been my experience that is the advocacy groups, it is the people living with disease and their advocates and their caretakers that know more about the disease and more about the science than the scientists and then the doctors. And so with that, I want to make sure to empower you all with how I go through this evaluation and triage when I'm looking at either investment opportunities or thinking about starting a program. What I try to do is ask myself, what modality should we use? Should I be trying to develop a small molecule that will work? Should I try a large molecule, an ASO, which I'll explain that one if you haven't come across it? Should I be thinking about gene editing? Should I be thinking about cell therapy, or is it AAV? I'll spend more time talking about AAV because I have a lot of cool data that I can show. But with this, I like to think about how these drugs interact or how they affect a system. And doing this at a really high level, small molecules, so we can think about them as pills. It doesn't necessarily mean they're just pills, but small molecules usually will modulate a pathway. So this is lots of different proteins that are interacting, and they'll modulate it by sticking to something and changing its shape or blocking an interaction. Large molecules, which usually those are a biologic, so a protein like an antibody, or even maybe the protein that's missing. Usually these are used to replace that missing protein or to perturb and block part of that pathway. And that's something that you'd be using over and over again. And ASO is an antisense oligonucleotide, which is a lot of words and a lot of syllables, for something that is a single strand of what looks like DNA that can get in the way of things to change the amount or level of expression of a given protein in a given pathway. So that allows us to change how much of another protein we're making. And gene editing, which is really exciting, is a way that we can try to address the genetic errors in a pathway directly by changing that blueprint, those A's, T's, C's, and G's that make up our DNA, that then give us the instructions to make proteins. And then the last one, and I put it last because it's the hardest one, is cell therapy. Cell therapy, what we're doing is we're making cells that do the job that isn't being done. So that might be that we replace disease cells, like in the case of Parkinson's, where that disease happens because we've actually lost specific neurons. And there are drugs that are in late phase trials putting those cells back in. Or it's where we're making cells that do a job that wouldn't be done otherwise. There are multiple approved therapies in cancer where we take cells and we give them a targeting sequence to go find that cancer, attack it, and kill it. And our immune system does do that, but this is supercharging that and making almost like a heat-seeking missile for that cancer. So in that way, cell therapy is replacing or putting in cells that are going to do a job to fix a pathway. And the last one is AAV, and I'm going to talk about it the most because, frankly, it gets used the most. In some cases, it actually makes sense to use. In other cases, folks may be just grabbing at it because it's what they know. And it's to that point that it's important to think about how we spend our time and energy because drug development is costly, not just in dollars, which is really hard to raise those money and put them forward, but I would argue in something much more important, which is our time. In theory, you can always raise more money. It is very difficult to find ways to get more time. And so for me, when I'm starting on a therapeutic drug development journey, after sitting down with a patient advocacy group and understanding more about what the unmet needs are, the thing I'm going through in my head is what is the right technology to bring in? What can we do? And does that actually make sense? Should that be our first thing or should that be our backup? If you want to present this maybe from the more business case side, one of the ways to think about it is if this disease makes sense to treat, and there are ways to treat it, other people are going to come in and try to do the same thing. So you should be trying to do it the smartest way, the easiest way. And I've put these in reverse order. Small molecules the easiest, and then going down becomes more complicated. AAV and what we do is different. And some of these slides are from Apatura, the company that I help lead. We develop new versions of this thing called AAV, which I'll share a slide on. And the way that we do that is we engineer them to work with specific receptors in the human body in order to help make the drug go to the places we're hoping it can go. We're able to do this because we've been working with our scientific founder, Ben Deverman, who has focused his career on developing AAVs. And we've put out some exciting data, but critically, and the thing that actually I'm most proud of, and I think the reason why Heidi and these bits is because we've tried to make sure that we focus on actually talking with and sharing information with rare disease advocacy groups, because we recognize that there's no company that's ever going to be able to work on everything. The only way to have that ever happen is make sure that we're willing to and able to share our information and share our materials and share our technologies. Pfizer is a huge company, but they can't work on everything. I don't know why one startup would be able to do anything different. AAV and this virus we're talking about is something that is out in nature. AAV is a single-stranded virus. It kind of looks like this, although it doesn't usually have labels on it when we find it in nature. When it's found in nature, it looks like this in that it has just two big genes in it. One is called rep, the other one's cap, and all that does is code for itself. So the information it normally contains says how to make more of itself. AAV is not associated with any disease. It does not cause any diseases. It just kind of hangs out. And what we've figured out how to do in gene therapy development is actually take out all these things. I realize that I'm pointing with my pointer, but I don't have my laser pointer out. So we take out all these things that are normally in there. And what we do is put in new information. So neither of my parents have degrees in biology, and I'm an only child, which means that they ask me a lot what I'm doing and what I've been doing throughout my career. And so I have absolutely no doubt that my parents probably know the most about gene therapy of anyone else's parents, unless they do gene therapy, unless they're doing that sort of research. And the way that I've talked with my mom and dad about this is I always think of AAV as providing kind of like a software update for cells. And what we're doing is we're putting new information that was maybe missing or incorrect, and we're giving a corrected version and dropping that off so that cells can read it. And the way that we do that, or the way that it happens, is that this virus gets close to a cell, it'll bind to a receptor, and that's just a protein that's grabbing it, and that'll allow it to come into the cell. Once it's in the cell, it'll jump out, and then it'll make its way into the nucleus where we hold on to all that information. It's kind of like thinking like we plugged in the USB drive and then we're downloading the file. Now it's on the hard drive. And it drops off that payload that it had. And it makes this little circle. We call this an episome because scientists always want to have special words for their things. This episome hangs out in the nucleus. It doesn't integrate into the DNA that we have. It's not doing that. It's just going to hang out with a little copy. And then we can start reading those instructions and going and making protein. Now, the thing that's interesting about AAV, and every virus, if you talk to virologists, have different cells that they target, but usually one of the big questions is whether or not they target cells that are actively dividing, that are doubling, or if they hit cells that are not dividing. And this is more about the biology of how this part works. And really importantly, AAV can drop off its information in cells both that are dividing and those cells that are non-dividing. And the way that it sets up its information and hangs out inside of that cell, the fact that it doesn't integrate means that if the cells that are targeted are rapidly dividing, we'll end up diluting out the virus over time. So what that means is that if we were looking for a therapeutic effect, and if in order to have that therapeutic effect, we'd need to be in those cells for a really long time, and those cells are rapidly dividing, we would then lose that outcome. And eventually we would see the disease state come back. Now, if the goal was to do something like gene editing that you only need to do once, maybe that would work. But in general, what we end up using AAV for is targeting specific cells that are long-lived or ones that aren't rapidly or actively dividing. Now, interestingly, the cells in our brains really are like that. They don't really rapidly divide. We have most of what we're going to have throughout our life when we're born. And the liver also, in general cases, is about stable. You go through a number of liver doublings as you're growing up. I think by the time you're about 12 or 13, you've had 6 or 7 of these. But the cells in your liver are quite long-lived. So if we think about diseases that are neurometabolic disorders, ones where it's both a disease of metabolism that also has impacts in the brain, and we need things happening in both our brain, and I'm pointing at my liver, or I'm attempting to, but I'm not on screen. Something like AAV might be the right choice to be able to drop off that information. There are a lot of different AAVs. So we call these things serotypes, or we call them capsids. And a lot of these have been found in nature, and then folks like me and others much smarter than me have tried to engineer better ones that do a better job at, say, going to this place but not this place, or vice versa. This chart is something that's out there and talked about a lot of, like, if you were just starting from scratch, which tool would you pull down from the shelf? Which one goes to which parts of the body more efficiently or effectively? There are multiple drugs that are approved now using AAV. There's one called Zolgensma, which uses AAV9, which is this one right here. It's not actually purple in real life, I promise. And that's for little babies with this disease that used to be life-limiting. Now, that drug has been dosed in more than 4,500 kids. SMA1 used to be one of the leading genetic causes of pediatric death, and that is no longer the case. It's no longer the case because of Zolgensma. It's also no longer the case because of 2 other drugs that were developed in and around the same time. One was an ASO and one was a small molecule. But Zolgensma is still consistently used, and is probably one of the most beautiful examples of using AAV for a utility like this. What we've done at Apertura is think about, what would it take to make these things better? And in most cases, the thing that we believe that would make it better is getting the virus to the right places in the body so that we can drop off that software update. And critically, the place where we felt like there was a large unmet need was being able to get things into the brain and cross the blood-brain barrier and drop off those software updates. So I'll show you a couple of slides on that. And just jumping right off, this one here is a mouse brain. And what you're seeing in the blue is like all those nuclei. So every cell has a nucleus, and we're seeing that. Now, in this one, you can see that there's green here as well. Lots of little dots of green. Each one of those little dots of green is one of those virus episomes I've dropped off. Now, this thing here on the left is AAV9. This is what's used in Zolgensma. When we use it IV, it's not very efficient at going to the brain. Especially when working in adult animals. These are mice that we're looking at. This one here is the first generation capsid that we have that uses the human transferrin receptor in order to hitch a ride and cross the blood-brain barrier and able to get there. And we're hitting basically every single cell that's in there. If I turned off the blue, which I don't have that slide anymore, I used to have it here back to back, Heidi. If I turned off that blue, you would see what looks like just the brain in the same way, because we are getting everywhere. The blood-brain barrier is something that keeps us safe. Everywhere that blood touches brain, we have the blood-brain barrier. It's not in one spot, it's everywhere. And our brains are made up of loads and loads and loads of cells. You might have heard people talk about or joke that we burn a lot of calories in our brain, and therefore, like, maybe if you do a crossword puzzle, it's the same as going out and running a mile. Well, last time I talked to my doctor, I'm still supposed to go out and go running. Can't use crossword puzzles instead. But critically, what it means is, like, all of our brain needs both oxygen to come in. We need to take out CO2 and garbage. We have other molecules, as I'm sure this community knows very well, that are important to get into the brain for energy and metabolism. One of the things that's really important to get there is iron. Iron doesn't just hang out on its own, though. It hangs out in this protein called transferrin. And transferrin is a really, really, really big protein. And the blood-brain barrier is there to protect our brains from bad things. Those bad things include bacteria, they include weird foreign proteins, they include viruses. And the way that we have to deal with iron is, like, iron on its own can be dangerous. That's why we shove it into this protein, into transferrin. Transferrin on its own can't get across the blood-brain barrier, even though it needs to. And what it does is it uses a protein receptor called transferrin receptor, or TFR1. And it grabs onto it and it hitches a ride. So transferrin receptor helps it go across this, like, cell barrier and cell membrane So it grabs it on one side and then brings it through and drops it off. And that process is called receptor-mediated transcytosis, which again, honestly, is scientists making up words to make them feel smart. Really what we're doing though is it's kind of like a ferry that we're jumping on and jumping out of. This is what the body does to get things across. Transferrin receptor is probably the most well-studied and well-understood of all the receptors for the blood-brain barrier. And it's one that is highly conserved in how it works across mammals. And the distance between blood to any cell in the brain, if you're you, if you're a mouse, if you're a monkey, a dog, a cat, a blue whale, it's always the same distance. It's always about 20 micron, which is about 2 cells apart, or 2 cells width apart. And this kind of makes sense because we need to make sure we get oxygen and food everywhere in the brain. And everywhere in the brain, we have the human transferrin, or we have the transferrin receptor being expressed. So what we're doing is we're taking advantage of that, and we're going to try to hitch a ride on this protein in order to get across. Now, we're not the only ones doing this. We're kind of cheating off of our big brothers and big sisters and all the homework that they've done. There are a couple of companies, JCR, Denali, and Roche being a few of them, that have either already developed drugs that are approved that use the transferrin receptor to help bring the drugs across the brain, or have things in late phase clinical trials like Denali and Roche that are doing exactly the same thing. JCR is a cool example here. They have a drug called Iscargo for a disease called Hunter syndrome that is a neurometabolic disorder, where they've put the enzyme that's missing onto the back of this antibody that uses the human transferrin receptor to cross. And that would be a really great way to do a lot of different things for a lot of different diseases. And the complexity and the thing they're lucky about in Hunter syndrome is that enzyme that they're bringing across also helps with cell targeting and get to the specific cells that need it the most. What we're saying when we're using AAV, though, is if we have something like the human transferrin receptor, we think that there are a lot of opportunities to unlock the opportunity to help in these different diseases that are everywhere in the brain or in specific spots in the brain, because everywhere in the brain has an interaction with blood. So what it would let us do is administer a product, not by doing brain surgery and sticking a needle in, but instead by just doing an IV infusion and then letting nature take its course. So our capsid, we call TFR1 CapX, would bind to the receptor. It doesn't displace transferrin, it just hangs out with it. And then it would cross the blood-brain barrier using that receptor in order to be dropped off. And we've done a lot of work to look at this. We have a published paper, actually, that earlier images I showed you is from a paper in Science that Ben published. We understand exactly how it binds to the receptor, where it binds to the receptor, even down to the amino acid that it's sticking to. And it's because of this that we've been able to find out that ours is human specific. It's only going to work with the human transferrin receptor. With this, the things that make us excited, we've done a lot of work to look in multiple animal species, ones that don't have the human transferrin receptor, so non-human primates or monkeys don't have it. They're not useful. We have actually a mouse that has the human transferrin receptor, and we've been able to use that to show the distribution of our virus. And we know from other drugs that have been developed that mice with the human transferrin receptor knocked in are representative of what it looks like when you use these drugs in people. And because we've seen so many other people move these things forward, we're buoyed by all of that information that they've supported. We have 2 programs that we're supporting right now, both for rare disease advocacy groups. One will have our first in human or first dosing happening in the second half of 2026. And there's a separate one that is projected to be in the first half of 2027. We're excited for this for a lot of reasons. One of the big ones for this capsid and being able to do this is that it allows us to use intravenous delivery. We think this is important because it makes it easier to make sure that folks have access to the product. It doesn't require fancy neurosurgery. It means that it's much more scalable so that once the drug is approved, it's easier to get it out to lots of folks. We don't have to then spend time training neurosurgeons on how to do a specific surgery. And neurosurgeons are really smart. They can absolutely do that. And there are a couple of really exciting programs in gene therapy that use a neurosurgery approach. The thing that I am excited for is for those to move forward. The thing that I'm scared about is we don't print new neurosurgeons very quickly. And once we get more drugs approved, it's going to be harder and harder and harder to keep rolling out new ones because there are only so many hours in the day to do administration of these products. So for us, we're trying to think about this again with that end of like patient 1000 in mind. We have a pipeline. This is usually when I'm doing the salesy bit, but just saying we have multiple things we're working on. Aperture is working directly on TSC1 and TSC2. These are rare genetic epilepsies. And then we're supporting these other programs. We have a number of partnerships, including one with the Rett Syndrome Research Trust and a few companies. The thing that makes me proud of what we're doing is the fact that we are actually trying to make sure that we talk to everyone in this, and we're not just shooting for Pfizer or for Merck or one of these big drug companies, because we realize that the place where innovation happens, and most formally, the reason why it happens is because of our advocacy groups and people saying like, we need this to move forward, and someone pounding on the table to move that ahead. So we're excited to be able to do both when moving things forward. I can show you some pictures of what our thing looks like and how it gets to the brain. These are mouse brains that we're looking at. Here's 89 again. This one is a mouse-specific capsid that a lot of people in neuroscience use called PHPED. It only works in mice. And then here's our capsid going everywhere. And the thing that I want to point out for folks, especially if you're not doing AAV development normally, is that the amount of virus we're using here, this number might not mean a lot, but that drug I was talking about, Zolgensma, that's already approved, this is 50 times lower dose than that. And we're reaching more than 80% of all the neurons in the cortex and more than 60% in the striatum, like deep structures across the brain. We've been able to show that we get a number of virus to those critical parts in the body. And then we also have virus in other parts. And this is something that we think about a lot when developing a gene therapy of where are we trying to go and where are we not trying to go, and thinking about the risks of that and like what software we're updating in what cells and in what ways. And so it's data like this that scientists and drug developers would use to think about where would this capsid make sense, what diseases would make sense, and then maybe what extra instructions do I need with that software update to make sure it's only read by certain cells. So am I going to make it be readable in every single cell and in every language that all of them can read? Or am I going to write it so that it's only in languages that maybe the brain can understand, or languages the brain and the liver can understand? When we're doing this and moving it forward, it's really those questions we're asking over and over and over again. I'll show you a little bit of data from the way that we've tried to put together a preclinical roadmap so that anyone and everyone can pick this up and do it themselves. Of course, like we're happy to be there to help. You don't need to do it on your own. But with that, we've taken the information and the learnings we've had and the interactions we've had with the FDA to be able to say, like, we know how to move this type of a drug forward. And we've gotten confirmation from the agency that using our capsid in the programs that we've been working on, we don't need to do additional non-human primate studies. Instead, we can use these mice that have the receptor knocked in, and we can use those as an equivalency to humans. We've also done work to say that we can leverage existing data if we've done things in disease models already to show that we could benefit them, that we can use that information and more quickly move forward, and we don't have to redo work. We've done that with dropping off just a software update. We've also done it with a complex form of gene editing that we've done in collaboration with Sonia Valab's group over at the Broad Institute. I can show you some of their data later on in this. Sonia Valab is a patient advocate and patient. She has prion disease. The surrogate approach, and what I was mentioning earlier, that we can take existing data and then bring it across. I'm not going to go through all the science on this because I know like my eyes glaze over it on this even. The thing that I want to get across is that we have alignment to say we can use a disease model and show that we can benefit it. And then we can separately work with this distribution model and show that if we can find the math between the two, we can move forward rather than needing to breed mice together over and over and over again to make more and more complex models that try to get us closer and closer. And the reason why we can do that is because we understand how these drugs work. They work because we're dropping off information and we're dropping off that software update. And we can actually look for that software update and understand how much of it was needed in order to have a therapeutic effect. And so this ends up getting simplified down to something where we could effectively, for most of these diseases, execute what looks like 4 separate studies in order to be able to move forward and through and answer all of those questions that I showed you earlier, those 7 questions. And in some cases, you can even collapse some of these data. So we can look at what it takes to benefit a diseased mouse in an efficacy study. You can look at what it takes to deliver and get things into a mouse at all, and if it's safe. And then after that, we can move over to our actual therapeutic capsid and therapeutic product in these knock-in mice to understand what that distribution looks like, and then convert over and run a toxicology study that then allows us to move forward into people. There are some programs, this is probably the longest path, there are some programs that we're supporting where you can quite literally kind of start right here in this efficacy study and then immediately move over to tox because you've been doing overlapping experiments. There are some groups that we're helping that are going from another capsid, and we're helping them transition over to ours. And there are some groups that have an existing program and they want a version 2.0, and we're helping with that. And all it is asking and answering the question of what information do we have from either cell lines or disease models or other efforts, and how do we then combine that information with the new stuff that we have to tell us how much virus we need to use, where do we need to get it, and how much needs to get there to have a therapeutic effect. It's really just those questions over and over again. And so as we think about it, we've gotten alignment on how to execute those studies. We've got an alignment on what doesn't make sense to do or redo in terms of work. And then we've set that precedent to understand that we don't need to spend extra time and energy to cross those animals. Even though it's exciting and it does check all those boxes, it takes a really long time and it doesn't actually give us that much more information. This is what's sufficient for meeting FDA's requirements. But thing that I remember and I try to make sure I teach my teams is that it's not just about satisfying the FDA. In my experience, there are about 5 different groups that you need to be talking to in this. And one of those that should always be thought of first are the patient advocacy groups and those folks that are actually going to be enrolling in the trial. And it's important that instead of just thinking about what are the boxes that we need to check for development, we ask, like, what are the pieces of data that we need to see for folks to have the comfort and confidence of volunteering themselves to be part of a clinical trial. So we have them. We also have the clinicians that would be running the trial. We have to make sure that they feel comfortable with the data that's being generated, the hospitals that are allowing the trial to occur there, and then the company or the advocacy group that's actually developing the drug has to have comfort. Then it's the FDA. So it's these 5 stakeholders that all matter and come together, which is why it's important that it's not just how do we develop a drug, but also what actually matters and what data and what models do we trust and have confidence in. This last bit, and then I'll be done so we can go through questions and answers or anything else like that, or just chat, is looking at prion disease. Prion disease is a fatal neurodegenerative disease that's caused by protein misfolding. Some people might be more familiar with something that's called mad cow disease. That's prion disease. And there's actually a genetic form of prion disease where people have a specific mutation that makes the protein much more prone to this misfolding. And once some of these proteins are misfolded, they actually tell their buddies to misfold as well. And they turn into these aggregates that eventually kill neurons. So this disease development and pathology is actually very, very, very well established. So you have the opportunity to stop this from happening, from intervening at any one of these steps. So you can edit the DNA to make that thing not be there anymore. You can try to get rid of the mRNA to eliminate the protein, which then means that you won't have this folded. And Sonia has been working in this field. She left her old job to go get a PhD, to go start a lab at the Broad Institute, and then devote herself to developing drugs for prion disease. She has a beautiful TED Talk on this. And it's through her work that there are a number of things that are actually moving forward. And one of those that's really exciting is an ASO. That ASO and this data I'm showing you here is in mice where they've actually purposely put in a mouse version of prion to seed the development of the pathology. What they've done here is they've administered the ASO either before any symptoms or before inoculation or prior to symptom onset. And what you're seeing here in the blue is the lifespan of these mice. What you're seeing here in black is the lifespan of the mice that weren't given that treatment. What you can see here is that if given prophylactically, so before any disease onset or pre-symptomatically, before anything that you can see, it significantly increases the lifespan of these mice. Whereas if the drug is given in the early symptomatic or late symptomatic stages, there's very little benefit. What this tells us, and what's interesting to note, is that if we can get rid of the protein that is going to also get misfolded, we don't get more activity, we don't get more cell death. So with that thought process, they developed a really complex way to do this inside of cells. They figured out a software update. Here's the little software update here that could get dropped off. And what it does is it recruits machinery inside of cells to label the DNA with methyl groups to turn off the ability to express the RNA, which would then make the protein. Sonia and the team published this work in Science, which is a very fancy journal. So if you're in academia, you know what I'm talking about. And if you've read a lot of papers, they're the ones that just say Science at the top, which I've always thought is a silly title for a journal. This work that they did is exciting because this is a new way to modify gene expression. And this does this without cutting any genes. It's just coming on and labeling them and making it harder for them to be read. So now what I'm going to show you is this is a mouse brain again. And this time what we're seeing, you see all these little white dots. These are all the little copies of mRNA for prion. Okay, and this is a healthy mouse, and we have expression of the RNA in both neurons and glial cells. So basically every single cell in the brain. And then what we're going to do is we're going to take that mouse and we're going to treat it with an AAV that has this software update targeting prion to inactivate that promoter. So this is an animal without anything administered. This is one that has. And that's not an issue with my slides. Like, it is just that quite literally at a very low dose run systemically, we're able to get virus all the way across the brain and turn off almost the entirety of all the copies. This is using the capsid that we're using therapeutically. This is using a mouse-specific capsid that we usually compare to. The thing that's amazing here is that this capsid here is usually thought to be like one of the best for neuroscience work. And what we're seeing in the data that we have is showing that TFR1 CAPX that actually uses the human transferrin receptor to get things across is significantly better and more potent. In fact, it is 10 times more potent at the same number of viruses getting into a cell and being able to knock down activity, which for us tells us that we can use less and less of the virus in order to get the same outcomes. The place where that makes me excited is when I'm thinking about all the really exciting academic work and publications of people, making sick mice and then finding ways to heal those sick mice. They're using things like PHPEB or AAV9. And if they can do it with that, then it's very feasible to then, if we're going after the CNS, use CAPX instead and have something that's humanly translationally relevant. Be used and bring that over. And not only can we do that, we're also going to be able to do it at a lower dose, a significantly lower dose. In order to have a therapeutic benefit in prion, we need to have about a 50% reduction. And that means that we're able to do that at these dose levels, even as low as 6E12 moving forward, which is really exciting to think about because less material going into a person is always going to be better than more material. I think my last slide is just like a call to action. I guess I'll leave this up for a second just to say, like, our goal is to move the field forward. We think that the way to move the field forward is making sure that people have access to things. It's not just about access to the right cells to be able to do the science, but actually access to the right tools to be able to do it. The thing that my team loves doing is spending time with folks and helping them think through how to do this themselves. And I think a big part of that is that these efforts in doing drug development, this is a joyful endeavor. It's hard not to be optimistic when you realize that you have the opportunity when you're doing research to just change our understanding about things fundamentally and go from a world where we didn't have that knowledge to where we do. And then eventually, if you have enough of that knowledge, actually move things forward into therapies. It's because of like Heidi reaching out or getting that first email and then getting to chat with Heidi that I've gotten to learn more and more and more about your field. And there's still so much for me to learn. So if you have questions, I'm not going to be able to get into like molecular mechanisms of creatine deficiencies super quickly. That's where I'm going to need like Heidi's help. And frankly, you guys know more about that than I do. But when thinking about drug development, and specifically when thinking about gene therapy or AAV, I'm happy to take time now to walk through questions and answers. I'm also happy to answer questions outside of this. So if you're uncomfortable asking something here, or if you're too shy, either you can message Heidi and Heidi can make sure that I get that information, or I shared my email earlier. It's asteinsapper@deerfield.com, or you can connect with me on LinkedIn. I'm always happy to chat about this stuff. Just need to know that you have the question. So Heidi, with that, I'll pause and then maybe give you prerogative of first questions and then open it up to everyone else.
Awesome. That was really, really great, Andrew. Thank you. I do have a couple of creatine deficiency specific questions, but I think these are in your wheelhouse. Would you happen to be familiar with how many times gene therapy for membrane protein has been attempted? Asking this obviously in relation to creatine transporter deficiency, and has it been successful? And there is the recent example of cystic fibrosis, which is a transporter, and it had failed. Are there some success examples out there?
So not yet in a drug that's approved in gene therapy, but we have precious few of those. I've worked on cystic fibrosis a couple of times. It's funny when you ask about like, are there examples of this working when it's a membrane protein? That doesn't even come into my calculus. So like when I'm thinking about it, the first question I have would be, do we know that it's for certain that protein? Which in this case, yes, we do. We know what it is that's at issue. And then I would usually ask like, do we know where this needs to be? And do we have examples of where we need to not have it? I think we know that for this as well. So in principle, I wouldn't expect this to be significantly different or harder than other things that we've done. Now, when making therapeutic protein that you use as the therapy itself, membrane proteins are extraordinarily complex and very hard and almost not feasible. The trick that we end up doing, like if I was going through that triage of like, what is the right modality? If you told me, oh, it's a membrane protein, I would say, oh, like we really need to pause here. Doing enzyme replacement therapy might be too complicated because those membrane proteins like being in like a really oily environment, and they don't really like water. That's why certain parts are outside of the membrane and certain parts are inside of the membrane. But when we give enzyme replacement therapy, everything's in water. It's going into your blood and then it goes and finds its way. And it's very hard to get it to be happy in both places. So instead, encoding that information in the software update, either inside of a virus or as mRNA, for instance, in a lipid nanoparticle, that would be much more feasible. But I wouldn't immediately have a concern, Heidi, on it being a membrane protein. That shouldn't change its delivery practice.
Okay, and you touched on my next question, knowing where we need it to express and where we need it not to express. Is there any way to regulate the capsid expression in certain regions of the brain? So, for example, if we didn't want SLC6A8 expression in a certain area.
So when we say things like area of the brain, sometimes area of the brain might mean certain cell types in certain areas, and sometimes it just really means certain areas. When it comes down to getting in the weeds there is a lot of work that has been done to look at the promoters. So these are the bits of genetic instructions that tell us how to read or where to read things to then go make them, where those can be used and co-opted when building your blueprint and that software. So in some cases, you can even use the standard one, which is only read in the standard cells, and kind of take advantage of that. Now, I haven't spent time to go look at like how big those promoters are, if there's work going on in that. There are existing promoters that folks have used. So there are a couple of standard ones. There's one called HSYN, which is human synapsin promoter. Synapsin, which sounds like synapse because scientists are not that creative, is a promoter that works in pretty much like every single neuron. So when people are doing neuroscience research and they want to see things in all neurons, they use HSYN. There are special types of neurons that are GABAergic, and there are specific promoters you can use to just express in those. So there are a number of tools in our toolbox that we can pull in. And then there are other tools we can pull in that say, don't read this in this place, or don't read this in that place. So in theory, you could actually construct something that gets you all the little logic circuits that you want to build there. I used to help lead research developing things like that. And I will tell you that the most complicated thing is not figuring out how to do that. It's figuring out what someone wants. Because if you're not aware of the disease that you could be working on, you don't know which parts to put together to go make that blueprint. You need someone. Actually, I used to explain this to folks as like when you're going to Subway and you're ordering a sandwich, like if you have every single sandwich making material, you could make an infinite number of different combinations. But if you don't know what someone wants, it's useless. So it's a lot of tools like this that if you have that biological understanding, you could get super specific. The work that I've been doing for a number of programs, one of the first questions I ask, and I do this asking it in animals, not disease animals, but usually healthy animals, is what happens if we don't select where this goes? What if we do just put this everywhere? And the reason why I ask that is because sometimes our knowledge or our understanding of the biology is flawed. It's flawed because people haven't asked the question, because people have assumed a specific answer. One of my mentors had said something along these lines, and I take it very much to heart, which is to remember that the first thing that we should be doing when working in drug development is to start with humility and recognize that like the proteins that we give names to and that we say do certain things, that's not what they do. That's one thing that we know that they do. It's not as though they're telling us their names or everything. We can't interview them. So I like to, as a first pass in these things, Heidi, usually just go express it everywhere and see, is that going to cause a problem? And sometimes that work's already been done. And then, you know, okay, cool, there is a problem. I need to change this and do that. The other versus starting with, in some cases, like dogma for that.
So that leads me right into my next question perfectly.
We do not plan this.
You know, trying to see where is this expressed and how well is it expressed and is it safe to express it everywhere, and you know, just learning all about this. You know, one of the advantages, obviously, of this vector is that you can skip the non-human primate testing.
Yeah.
But if you skip the non-human primate testing, how do you assure yourself about, you know, the distribution and also especially the optimal dose and toxicology and all of that?
Questions. So I think it's really important to recognize, like, when we're doing this, we're not cutting corners. So when thinking, I'm going to try to think about the order to answer that question. I think the first one to start with is dose, because I think this one matters a lot. We know that our number is 1-to-1. And the reason why we know that it's 1-to-1 is that we know that when you scale up a brain, so my brain, a mouse brain, my cat's brain, my mom's brain, my dad's brain, in all cases, it's always the same distance from blood to all the cells in the brain. So that doesn't scale. It's the same. So if we need 1 times 10 to the 13th viral genomes per kilogram in that mouse, we're going to need about 1 times 10 to the 13th viral genomes per kilogram in the human. And we've seen that in work using products that use the human transferrin receptor to cross the blood-brain barrier. So Iscargo, the drug that was developed and approved for Hunter syndrome, is approved in Japan. You can go download the packet
on the Japanese FDA's website. It's PMDA is the name of the regulatory body. And they have a copy of all of these packets, either in English or in Japanese. And I read moderately good English, so I downloaded the English copy. And what you can see is that in the studies that they use to look at the dosing administration, and then what ended up being used in people, it's actually the mice that were predictive of human outcomes, not the non-human primates. Now, they did execute studies in non-human primates. And the reason why the non-human primates weren't predictive in the same way is that they were going after the human transferrin receptor, not the non-human primate transferrin receptor, which in and of itself kind of changes how it associates and whether or not it'll get across. So that's on the dosing side. When you ask the question about like off targets and like that scaling, that actually goes along the same way. When we're using blood as the route of administration, all organs effectively see it the same way, even when we scale up, even though I know a mouse is much, 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 much smaller. Now, in reality, when folks are doing other forms of AAV administration, they'll do things. There are a couple of acronyms folks will use. One is IT, or intrathecal, that's like into the spinal cord, into that space there. Another one is ICV, which is intracerebroventricular, which is it's brain surgery, and then you're putting it in these fluid-filled spaces in the brain called ventricles. There's another one called ICM, or intracisterna magna, which is a spot back here, again, CSF. Now, like those, that fluid that covers the brain, people are saying like, oh, it's that much closer to the brain, we should go use that. It's there directly. In reality, the scales and distances that we have to cross when we're in a mouse versus a non-human primate and then a person are very, very, very different, and they're not linear. In fact, the scaling factor is about 1,200 to 1,500, depending on how you do the math, between a mouse and a human. It's still about 500-fold when you go from non-human primate to a human. And it's not perfectly predictive of those. So with this and across the board, when thinking about animals and scaling, the thing that I like to think about for drug exposure is how does the drug get there? And how does that system change, if at all, across them? I think you also asked a question about safety. So I present on this often. And there was recently an unfortunate death in a clinical trial using an old capsid. Did not use the human transferrin receptor. It used another receptor that company has not specified which one. I mean, it's terrible. I was rooting for them. We've been rooting for everyone because we want this to work. We want it to work for everyone. The work that they did, their work was done in non-human primates initially. They actually found their capsid using non-human primates to develop it. They put out all their toxicology safety data at ASGCT at the big gene therapy conference. It's a conference where I met Heidi for the first time. And they didn't have any issues. And I think this also reminds us, and it's been true across other gene therapy programs as well, there is no model that predicts human safety. No. There's no model that perfectly predicts it. In fact, humans don't even predict human safety perfectly. And the reason why I say that is that there have been unfortunate deaths in clinical trials where we've not been able to predict what characteristics should tell us don't work with that person, like don't put them in the trial, that's going to be dangerous, because we don't know how to evaluate those outcomes yet, because we don't understand that science completely. It's one of the reasons why it's really important when executing clinical trials to collect all that data and that information, because it's from those unknowns that people go, there's something that we're missing. The way that we're thinking about this is not complete because we weren't able to predict this. And then they actually go backwards and try to build translationally relevant models that help answer those questions. The two largest safety events, other than the capsid event, are for something called TMA, which is effectively ends up being like buildup that causes bleeding. And you see that in the liver. That's in no small part a result of having antibodies against the virus early and beforehand. But knowing who is going to respond and who isn't there's no perfect prediction for that. You know, there are things that we can do in terms of using medications prior to joining the trial that mitigate the risk of that. And those are used in general across things. This is what came up in the Sarepta trial. The other is something called HLH, which I'm going to forget what the acronym stands for, and I really apologize. HLH is a different syndrome that happens from the innate immune system. And we also, again, cannot predict which people will and won't respond. There's some guesses of why this is the case, but there are medications that can be used prior to administration and in the case where this occurs to mitigate those risks. This, again, is the reason why it's really important when doing drug development to be working with hospitals and medical centers that have a lot of understanding and experience, not just in gene therapy, but across the board. It's something that as we were putting together Apertura as a company and we were developing these technologies, we thought it was really important that we do everything we can to explain and put out information about what we have so that if anyone ends up using it, they have as much information as possible to think through all the things that they could be doing to mitigate risk. And if I sound like I'm being weird in the words that I use, there's a reason. I don't talk about reducing risk. And that's because it's not actually possible to reduce risk. Risk exists. It just is there. You don't reduce risks, you mitigate risks. You find ways that should those bad things occur, you can do things to remove or eliminate harm to whatever degree you can. I like that framework and I like that thinking because I've been asked the question, after the recent unfortunate trial death, does that mean that this is riskier? And the answer is no. The risk has always been the same. It's static. The thing that has changed is people's uncertainty about whether or not things would occur. I've spent the last probably 6 weeks, since that happened, speaking to all of our partners, to speaking to clinicians across the board, to many patient advocacy groups, presenting. Actually, I'm at the Foundation for Angelman Syndrome Therapeutics right now. I'm calling in from a hotel. I left the conference so I could come give this talk. I'm going to be giving a talk tomorrow. Wish me luck. My slides are done. And one of the things I've highlighted there, and I will continue to highlight, is we understand how our system works. We know that there are hundreds of pediatric and geriatric patients that have been treated with molecules that engage the transferrin receptor, and there have been no safety outcomes in those patients. Critically, we've never done it with a virus. And as a result of never having been done in the virus, there's still a risk. But with that, and I think all the clinicians we've spoken to and our partners agree that at least we know how it works. We know that the receptor we're using is meant to be doing this job. We're not tricking something else into doing something else that it doesn't normally do. And that way we can go in with as much information as possible. We know that everyone in the human population has this part of the transferrin receptor. There's no variation right there, which also helps make sure that this is more accessible. But when it comes down to it, drug development is terrifying. And the only system that really matters when we're moving this forward is humans. And that's not to say be cavalier and just go jump to doing clinical trials. I say that because it's a reminder that no matter how much work we do, no matter how many studies and how many systems that we work in, the only data that's going to be meaningful for getting a drug approved and making sure as many people can get access as possible is going to be data in humans. And that is not going to change anytime soon. We can't AI our way out of this. We don't know enough about these systems in order to be able to understand how to build models for them. And so it's with that when I'm thinking through programs and doing drug development research or advising companies or groups on this, the thing that I ask myself over and over again is, does this answer the questions I need to ask in order to move into people? And does it give me an understanding of the safety and the potential efficacy of the product? Because if it's something outside of those, that's not actually moving it forward. Then it's probably more of a vanity exercise for me of trying to prove a point on some nerdy thing and not actually doing drug development. And I think that's, it's actually very easy to lose that thread when you're going deep and doing this work.
So there's a lot of talk about, building a platform. Mm-hmm. Once you've got a platform, you've got a fast track to mm-hmm. get to clinical trial. And, talking today and talking about, where does the gene go, and it's really more related to the promoter and in the plasmid, not the capsid. How do you feel about that concept of like there's a platform and it fast tracks to people? Because it sounds a little scary, and I think where we need our gene delivered is not necessarily the same as another disease, and how much we need. And it kind of seems like a too good to be true concept. What do you think?
I think that it's a concept that's poorly described and discussed. So the idea of a platform designation is about reusing the tools and technologies when it makes sense to do so. It doesn't mean that you get to skip things. It doesn't mean that something is, and I really hate this phrase, like de-risked, like, oh, it's totally fine no matter what in all cases. That could not be less true. What I like to think of it of instead is the places where that can be used is to tell you what not to do, right? So if I already know for a given capsid, for instance, where you would be leveraging that platform information, if I already know all the places it goes and all the places it doesn't go, I don't need to be spending extra time, energy, and money going to those places I know it doesn't go and checking, like, does it still not go here? Does it still not go here? Does it still not go here? And that might sound like a silly thing, but finding out what we don't need to be looking at, I think, is almost more useful because it's in those places that we can get distracted. Changes in promoter, very common, and it helps. Maybe this analogy will work. This is the one that my mom liked the most. My mom's an artist. She's an author. And when I was telling her about these novel capsids and their distribution, she said, oh, like, this is knowing what your canvas is. I went like, yes, exactly. It's where we could be. It's what we could be doing things on. And then it's up to us to decide what parts of it we're painting. And that's closer to like what promoters we're using. And then like what we're painting it with and what is that thing? That's actually the gene that we're putting in. And so with that, like, if you know that you have a consistent canvas that you're working with, you can then think about which diseases make sense or a one-to-one application. There are diseases that are extraordinarily closely related to each other. Like the two proteins interact on the same pathway, take your pick. They're completely like behave differently. And you can't just assume. So with that, I think it's going to be important, regardless of platform designation or not, to always enter into these things with humility and say, like, we've never done this thing before. Given that we've never done it before, what do we need to look at? And if we already know stuff doesn't go to certain places, we know it doesn't go to certain places. Maybe we can spend less time on that and more time on the other thing.
Yeah, great. Well, while we have a couple more minutes, if anybody wants to come off mute and ask a question or put it in the chat, while we have Andrew before the Angelman community takes him back. I think I just brought that up, and please, anybody that wants to ask a question, just jump in and interrupt me, please. I brought that up just to highlight, I think like your talk is very promising and this technology is very promising, and I don't want to leave everyone going, well, why isn't gene therapy ready to go tomorrow? Like sounds like usually you put SLC6A8 in there and we're good to go, right? And it's a capsid, and there is still the plasmid that's in it, and how that works in the body and how it distributes and what the right dose is. And there's still a lot of work to be done. And thankfully, there are a lot of different folks taking shots on this goal and looking at different capsids and exploring these ideas. We have a great scientific community who is very aware of these things in case this is also coming up in people's minds. So we've got a strong team, but it's still a very tedious and important process to make sure that the right gene therapy is given to patients and does no harm.
Yeah, I mean, that's the goal. So I've had the privilege of working on over 80 different programs in cell and gene therapy, from the painfully preclinical, super duper duper early, to commercial readiness and launch. And the only commonality across all of them is that it's always terrifying, because you're part of a team making decisions in effect for so many people. And there are so many different combinations and questions to ask and things to move forward on. And with that, it's for that reason it behooves us to make sure that we're doing it the right way. And the only way I know to know that you're doing things the right way is to know what we need to know. And there are examples that you might have seen in the media that are super exciting, like baby KJ and the gene editing effort for the adorable baby boy. And that stuff's awesome. I'm so excited to see that move forward. It's unfortunate, though, that that's not something that we can do for the brain. And that's because we can't deliver like that right now. You end up needing a tool like AAV to get there. And the baby KJ story is super exciting, but it did take them actually multiple doses to figure out how to get it at the right level to have an outcome. And when we use something like AAV, we only get the chance to dose once. And the reason for that, at least right now, the reason for that is like when we dose, we develop antibodies against the virus, just like we do for vaccines. And as a result, it would not be safe to administer to a person the second time. Now, there are other people that are working on medicines that will hopefully make those things go away, so we can look at that. But it actually raises those stakes, and it makes it even more important that we know that we're going in with something that can have the opportunity of being efficacious. Not just about being safe, but know that we're using enough of this to make sure that it has a chance of being efficacious. Versus when we do small molecules, we usually will start with a dose that's sub-therapeutic and then work our way up to find a therapeutic dose, because we can just turn that dial back and forth pretty safely. That's different in gene therapy. It's actually much closer to developing drugs in oncology, where you really get that one shot on goal for a patient.
Right. Well, I realized that I did not share the slides earlier. I'm going to fly through these really quickly, just to give everybody the follow-up of what I said. And Andrew, thank you so much. That was so informative, and you covered a lot, and I learned a lot. So I appreciate it. Rare Affair. This is the event that is happening in January 11th in San Francisco, that is helping to fund our symposium. And if you are in the Bay Area, we would love for you to join us. And Holiday Heroes, this is our annual campaign to fund research from November 1st through the end of the campaign, which is, we say December 31st, but we always extend it. So we'll just say it's January 31st. We restrict every dollar to research, and we have a lot going on in research right now. We've raised $1.5 million of our $5 million goal. So this is the year to get on board, and we have so many tools to help you. Please reach out. Please join our wall of champions in the Momentum League who are making a small donation each month to help us keep going. And join us January 16th for potty training for kiddos in our community with a specialist who goes to your home and has sat in on this event for multiple days and knows exactly what it is that you and your child need to hear and need to practice in order to accomplish this task. So with that, thank you again, Andrew, and everybody who joined, and have an awesome weekend.
Thank you so much, Heidi. And I shared a set of links if anyone wants them for all of our non-confidential slides. So if you have questions, you can grab those, and I can make sure that Heidi has them too. Always happy to talk. And thank you all so much. I'm so excited for this. And you guys do such a good job on the science education. I've been pointing other groups at you guys as an example of just doing this. It's so impressive. And I know it's so much work, but seriously, hats off to you guys.
Thank you, Andrew. Thanks for educating us.
Bye.
Bye.